also Crazy Sax. Yeah. Epic Sax guy. Wait, what is the song called? I like it.
It's Ghost in the Shell.
They're going to be back shortly, so why don't you guys sit and
Okay.
while you're dancing.
This is a great song.
Good luck, guys.
Okay, the third presentation is by Edward Wang Wayne Shu and Justin Yuan from the Troy High School in Troy, Michigan, entitled MicroRNA 17 to 92 Cluster Mediates Sonic Hedgehog-Induced Neurogenesis in Neural Stem Cells After Stroke. Come on.
Hi, ladies and gentlemen. My name is Edgar Wang,
My name is Justin Yuan.
And I'm Wayne Shu, and we're from Troy High School.
and we're presenting our project on MicroRNA 1792 cluster mediates sonic hedgehog-induced neurogenesis in neural stem cells after stroke. So basically, our research focused on the affliction of stroke, and stroke is a leading cause of mortality worldwide. It affects over 750,000 people in the US alone per year, and often the neurological deficits lead to permanent disorder that can't be recovered. And as you can see from the graph, the dark purple regions are hundreds of thousands of deaths, 100 to 2,000, 200,000, and this is within a 6-year period in just the US. So it's actually a very big problem in the US. And currently there's only one FDA-approved drug, which is tissue plasminogen activator, and this basically breaks apart the clot, but it can only be applied from 4.5 hours after the onset of stroke. After that, it has a chance of hemorrhage, so it's limited to only 5% of patients. Therefore, there needs to be a new therapy developed that can treat a greater number of people within a greater time period. So that brings us to neurogenesis. Neurogenesis is a relatively new science, and it's actually the birth of new brain cells. And it takes place primarily in the dentate gyrus of the hippocampus and the subventricular zone. And it's actually enhanced after stroke. So it's sort of like the brain's natural recovery mechanism of trying to recover from the damage.
Neurogenesis works in a 4-step process. First, it proliferates, then it migrates to area of damage, such as the ischemic striatum. They differentiate into cells such as the astrocytes, oligodendrocytes, and they integrate themselves into the pre-existing circuitry. Now, these newly generated neurons have shown functional properties of the neurons that were already in the brain. And they're involved in learning, spatial memory, long-term memory. And what's particularly exciting about this is that prior to neurogenesis, treatment of stroke has been mainly focused on neuroprotection and the reduction of the cerebral damage. But what we're saying is that the damage has been done. How do we help the person recover? With neurogenesis, with the neurons being born again, the person does have a chance for them to integrate, the neurons integrate back into the circuitry, helping the patient recover. Now, how do we enhance neurogenesis after stroke? First, we know that the approach needs to enhance cell proliferation, and it has to be related to the way stroke induces neurogenesis. And then we look to microRNAs, which might be a possible therapeutic target. MicroRNAs themselves are 21 to 25 nucleotide long RNA molecules. They're involved in neurocellular processes such as growth and development and regulation of apoptosis. And they can regulate gene expression through translational repression by the RNA-induced silencing complex. And they were discovered in 1993 by Dr. Victor Ambrose, who just so happens to be amongst our panel of judges. Now, of the thousands of microRNAs, why did we choose the 1792 cluster? Well, first, it's a polycistrine made of 6 microRNAs, which are transcribed coordinately, so they all have very similar function. And microRNA 1792 cluster has been proven to have strong angiogenic properties, such as in tumor growth. And it has been proven to be related to sonic hedgehog, which is a neuroregenerative protein in medulloblastomas, and so that there might be a possibility that 1792 also is related to neurogenesis.
So sonic hedgehog is basically a member of the hedgehog protein family, and it's also involved in a lot of processes despite its frivolous name. It's involved in neuronal cell differentiation, stem cell division, neurogenesis, and cell development. And basically, it can target a wide array of processes. But the molecular mechanisms underlying its neurogenesis in stroke haven't been discovered yet, so that raises the possibility that microRNA 1792 cluster could be the mediator of sonic hedgehog-induced neurogenesis. So we ask ourselves, is microRNA involved in the sonic hedgehog-induced neurogenesis pathway, and whether 1792 cluster can enhance neurogenesis by itself?
So for experimental design, we first want to establish whether microRNA 1792 cluster is related to stroke, and we first use an animal model of stroke with the middle cerebral artery occlusion. And after that, we test the SVZ cells to see the increased levels of microRNA 1792 cluster. We have an SVZ cell culture, and we extract the SVZ cells using laser capture microdissection and a dissecting microscope. And we use them for various experiments, and we also use a neurosphere assay to test for the biological functions of the neural stem cells. We use quantitative real-time reverse transcriptase PCR to measure the microRNA 1792 cluster expression levels. We use immunocytochemistry to test for the cell proliferation using a BRDU marker, and we also use Western blot to test for the target protein expression. As you can see from here, this is a picture of our laser capture microdissection. After stroke, we see an increase in folds of the microRNA 1792 cluster, which leads to speculation that microRNA 1792 cluster is indeed related to stroke.
So now that we've established that the microRNA 1792 is related to stroke, we question whether it's affected by sonic hedgehog. So first, we incubate cells in SHH for 24 hours to let them grow. Then we use quantitative real-time reverse transcriptase, PCR, to test the expression of microRNA cluster levels. Then we block sonic hedgehog signaling pathway, and we test downstream proteins via Western blot. So we implant the osmotic pump, which is a mini microosmotic pump, into the rat's brain, ipsilateral to ischemia. The osmotic pump is filled with sonic hedgehog, which diffuses through osmosis into the rat's brain. And so, as you can see through the chart, that microRNA levels have been shown to increase after the cells were treated with SHH. So we conjecture that if sonic hedgehog can increase microRNA levels, then logically, if we inhibit it, microRNA levels should decrease. So just to make sure that the two truly are related, we tested sonic hedgehog transfected cells with silencing RNA to test against sonic hedgehog. And as you can see, microRNA levels have decreased.
So how does this SHH mechanism, the signaling pathway, work? Well, we first have the sonic hedgehog protein, which attaches to a receptor called patched. And that releases a transmembrane protein called smoothin, and that leads to a cascade of functions which lead to transcription factors. So another way to test for whether this pathway really does affect microRNA 1792 clusters that we use cyclopamine, which is a known pharmacological inhibitor of smoothin, to block the pathway. And when we block the pathway, we see from our results that microRNA 1792 levels are decreased in folds, as opposed to not blocking the signaling pathway into our control.
So one of the downstream proteins of sonic hedgehog is NMAC. And as you can see, after we add sonic hedgehog, NMAX accordingly increased, and after cyclopamine is attenuated. So one of the target genes of NMAC is actually 1792 cluster. So it's very likely that sonic hedgehog targets NMAC to induce expression of 1792 cluster. So now that this link has been established, we want to see whether 1792 cluster by itself can affect neurogenesis. So we basically transfect cells with the gene for 1792 cluster, then we use the neurosphere assay and immunocytochemistry to test for cell proliferation. Now, this finding was the most interesting to me. So basically, we use BRDU, which is a marker of cell proliferation, and DAPI, which is a marker of total cells. And basically, the ratio of BRDU to DAPI, as in the merged picture, should show the ratio of proliferating cells to total cells. So what's really exciting is that after the 1792 cluster gene is transfected, there's almost a twofold increase in cell proliferation. So it's a very potent inducer of neurogenesis.
So we also want to find out how microRNA 1792 cluster how it affects enhanced self-proliferation. So we checked for the putative targets of microRNA 1792 cluster, and the target should explain why we have an increase in proliferation. And we performed Western blot on the target protein, and the protein that we looked at was P10. And P10 is a known cell proliferation suppressor. So when we use microRNA 1792 cluster, we see a decrease in P10. So logically, we can speculate that microRNA 1792 cluster can target P10 in order to increase cell proliferation.
To date, microRNA levels clusters have not been investigated after stroke, but for the first time, we haven't tested these effects, and we conclude that after stroke, microRNA levels have increased. And sonic hedgehog-induced neurogenesis is mediated by microRNA 1792 cluster. So therefore, the cluster mediates cell proliferation, an increase in cell proliferation in neural stem cells after stroke.
So what's cool about the 1792 cluster is that it could be a viable alternative to stem cell transplants. Because stem cell transplants are from foreign donor material, they may induce cancerous growths, but 1792 cluster works with the host cells, so this possibility is more likely precluded. And in the future, we would want to do drug testing with possibly like drug liposome deliveries with lentiviral vectors, which have already been tested in other microRNA research, so this is a very possible method to use.
So using microRNA 1792 cluster, if we can find a way to harness the power of neurogenesis and amplify it, then this could lead to a great recovery for the many people who are affected by stroke. And really, this isn't just limited to stroke. We use a stroke model, but for neurodegenerative diseases such as Alzheimer's, Parkinson's, and traumatic brain injury. These all are characterized by a net loss of neurons and also an impairment of neurogenesis. So microRNA 1792 cluster provides a great therapeutic target for gene therapy, and despite being such a small micro type of thing that we can use, microRNA 1792 cluster could have huge impacts for all those who are inflicted by all these neurodegenerative diseases. So we really hope that our research on microRNA 1792 cluster and neurogenesis can really be used in future clinical therapies to help better the lives of all those who are inflicted with neurodegenerative diseases.
And finally, we'd like to acknowledge our mentors, Dr. Chopp, Dr. Liu, Dr. Zhang, for being so patient with us this summer and helping us through our project, the Henry Ford Hospital Neurology Research Department for hosting us, our lovely parents, the Siemens Foundation for giving us this opportunity, George Washington University for hosting us this weekend, and our panel of judges for taking the time to listen to us. Thank you, everyone.